I'd like to welcome all the audience members to today's symposium in the series Emerging Topics in Biomolecular Magnetic Resonance. I'd like to thank the co-organizers who are listed here: Lauren Andreas, Stefan Glogler, Christian Griesinger, Oscar Miette, Mei Hong, myself, although I do very little, and Marcus Weckstetter. The sessions subscribe to the Gordon Research Conference policy, so please don't record audio or video yourself on your own devices. The lectures are recorded by us upon speakers' consent and then are made available at the ICMR BS YouTube channel, and the link to that channel is shown below. You can also find the link by going to the website for this seminar series, and the website URL is emergingnmr.org as shown at the top. Please type your questions into the Q&A. Don't type questions into the chat. That's not being monitored. If you want to be unmuted to ask a question, use the raise your hand function, and then we can unmute you. The introducers for each session will read the questions at the end of the lecture to the speaker, who will then answer them. So with that, I'd like to introduce today's first speaker, Francesca Massi, who is Associate Professor at the University of Massachusetts Medical School. It's a particular pleasure to introduce Francesca because she was a postdoc in my laboratory from about 2002 to 2007. Prior to coming to my laboratory, she received undergraduate training in Italy, in Rome, where she also grew up, and then moved to John Straub's lab at Boston University for graduate school, where she was trained in computational biology, doing MD simulations in Alzheimer's systems and other systems. So she came to my lab not having done any NMR spectroscopy. We had kind of a deal. She would teach us to do MD simulations and we would teach her to learn NMR. I'm afraid she learned much more NMR than we learned MD simulations. So we're constantly calling her up for advice now, even more than a decade later. One of the, I think, unfortunate parts of running a group is I always believe that the people who have left have really just gone out for coffee, and they're going to come back in a few minutes and we'll continue the productive and interesting conversations we had when they were in the lab. And so it's hard for me to believe that Francesca has been out for coffee for 13 years. On the other hand, one of the really fantastic things about having a group is you get to see the great things that people go on to do when they set up their own laboratories, and you have a sense that your own small contributions to science will go on in the future because of the people that you've trained. So it's fantastic for me to have the opportunity to introduce Francesca. As you'll see, she's continued to apply NMR and molecular dynamics simulations to problems in structural biology with a particular interest in protein RNA interactions. And with that, I'll let Francesca tell you about her work.
Thank you for the introduction. I'm going to share my screen now. Okay, so thank you for the introduction, Art, of course, and thank you for the kind invitation that gives me the opportunities to tell you about our work on a family of proteins that RNA binding protein that have tandem zinc finger domain. They use a tandem zinc finger domain to recognize RNA. So these proteins are important. because they're involved in post-transcriptional regulation. So what do I mean? Well, what is post-transcriptional regulation? So this is all the regulatory steps that occur after a gene is transcribed into RNA. And then, of course, there are several steps that lead to the as I said, many of these steps happen in the nucleus, and they until the mature mRNA is formed. And then once the mature mRNA is formed, it's exported into the cytoplasm, where it can interact with RNA binding proteins that will affect the translational efficiency of that RNA, the stability of that RNA, the localization in the cytoplasm or in subcellular localization, and sometimes it's sequestration into these membraneless organelles, this liquid liquid-like condensate. And ultimately, post-transcriptional regulation, we can think about it also in protein modification. Once the mRNA is translated into the protein, it can be further modified, and that can impact the activity, the localization, the stability of that protein. The proteins that I will tell you about today, they are involved in all the steps that can occur where mRNA is regulated in the cytoplasm, so they can impact translational control, the mRNA stability, mRNA localization, and mRNA sequestration. So in the first part of the talk, I will tell you about our studies of three human proteins. And then in the second part, I will tell you some newer data that we have on other proteins from this family that are from C. elegans. I will go pretty quickly through the first part as has been, as it's older and has been published. So as I said, we started working on this family of protein, and we focus on three human proteins of the TTP family. So TTP is also known as ZFP36 or tristratoproline, and this protein recognizes A-rich elements that are located on the 3' UTR of the mRNA. Once the protein binds the RNA gets degraded. As I mentioned, there are two more members of this small family of proteins, in addition to TTP, and they all share a very similar architecture. There is an RNA binding domain that is in the middle, and then an N and C terminal domain. The N and C terminal domain are both low sequence complexity, so they're likely to be unstructured. The RNA binding domain is a tandem zinc finger domain, which means that there are two zinc finger, and the zinc in each zinc finger is coordinated through 3 cysteines, CCC, and then 1 histidine, that's the H. Peter Wright actually had solved the structure for one of these proteins, TC11D, when it was bound to its cognate A-rich sequence. And it was a novel fold. As you can see, the 2 zinc fingers have very little secondary structural element, but they're highly symmetric in their structure. So the two structures can be overlapped, and then they're connected through a flexible linker. When we started working on this, and this is actually the work of a student in my laboratory that she has, and she has graduated since. What she realized was that despite the fact that the sequence homology in the 3 tandems in finger domain was extremely high, so TC11B and TC11D are nearly identical, TTP is about 70% identical, they have different structure in solution. In fact, in the absence of RNA, while in TC11B and TC11D, both zinc finger are nicely folded in one unique structure where the zinc is coordinated in each zinc finger. In TTP, only the first zinc finger is stably binding zinc. The second zinc finger is actually sampling many different states. So here I represent it as a fully unfolded protein, although, given the fact that in the NMR spectrum, we didn't have any signal from the second zinc finger, that indicates that the second zinc finger is in a molten globule. So it's probably sampling many different states, but what we know is that it doesn't bind zinc tightly. But regardless, once you add RNA, and if you have enough zinc in solution, TTP binds RNA, and it actually binds RNA with a similar affinity to TC11D and TC11B. So we were very curious to understand how come, given the the high sequence homology, how could one protein be a lot, be stable, structurally stable, and the other was not? And so here we use TC11B and TC11D. We did a lot of mutagenesis studies, and then my student Laura was able to identify 3 residues that alone can determine if the second zinc finger is stably structured or not. And in fact, if you switch these three residues between the 2 tandem zinc finger domain, you can get the second finger to be folded or not. And so then once we identified these 3 structures, we actually looked at the sequence alignment from the tandem zinc finger domain of TC11D orthologs among different vertebrate, and we see that these 3 residue HTI are conserved in all of them. But we're very surprised when we actually look at TTP that TTP has these the 3 residues that are associated to not so structured state in mammals, while all non-mammalian TTPs have the same 3 residues that are observed in TC11D, and so we can assume that those are all structure. And so this was a pretty remarkable and surprising result to see, because that means that TTP has evolved to be less structure in the second zinc finger relatively recently in evolution. But we still couldn't figure out what was so special about these 3 residues. One thing I haven't told you is that these 3 residues are located in the very short alpha helix. It's what the only secondary structural element that really characterizes this structure. And we couldn't figure out why the HTI could stabilize the structure of the finger while the other one, the YLQ, don't. And so what this is the work of my student Davide Tavella, who also recently graduated, and what he used computational a computational approach. So he ran molecular dynamic simulation, and what he observed is that when the finger is folded nicely, the histidine is that coordinate the zinc. It's stable in one rotameric state that is compatible with zinc binding. But in the protein, but sometimes in TTP, this histidine switches to a different rotameric state that is incompatible with binding. And so what Davide observed is that actually what keeps the histidine stable in the rotameric states, compatible with zinc binding, is actually a phenylalanine. And this phenylalanine is right next to these 3 residues that make the difference in the structure. And so what Davide observed is that what is really different between the structure of the 2 proteins is that the threonine in TC11D can form a hydrogen bond with the carbonyl group of the 3 residues before. And this hydrogen bond is enough to introduce a small kink in the alpha helix. Of course, it's a super short alpha helix, but it's enough to position that phenylalanine in a different place where it can either really effectively stabilize the conformation of that histidine or not so much. And so this was, so he came up with this model, and of course this was based on computational approaches. So he wanted to validate in, using mutagenesis. And so what Davide did was he first mutated the phenylalanine that is meant to stabilize that histidine. So if his model was correct, then he would expect that the structure of the zinc finger to be destabilized. And so he took TC11D, he made that mutation, and what he observed, in fact, is that the second zinc finger in TC11D, now it's unfolded. And even upon addition of RNA, that zinc finger cannot fold. So that phenylalanine is really critical to stabilize the structure. And just to give you a sense, you might think that phenylalanine, it's a small protein. If it's in a hydrophobic core, it might actually destabilize the fold of the protein, but this is not the case, because actually that phenylalanine is partially solvent exposed and is just there to stabilize that histidine, to interact with that histidine. So then actually we looked at all the sequences. Well, we downloaded from about 15,000 protein, that contain a CCCH zinc finger. It didn't have to be in a tandem zinc finger. And what we observed is that in 96% of these 96% of this protein at that position have an aromatic group. It doesn't have to be a phenylalanine, but it needs to be an aromatic group. And so we think that an aromatic residue in that position is essential to support the structure of the zinc finger. The second thing that Davide wanted to test was if this hydrogen bond between the threonine that affected the conformation, the position of the phenylalanine was essential. And so what he did is that he made a mutation, and in his mutation, and so he mutated the threonine to a leucine, and he mutated in TTP the leucine to a threonine. And then what he observed is that at that point, the proteins fold. So the presence of just that amino acid is sufficient to fold the tandem zinc finger. So we had actually stopped a little too early. It wasn't 3 residues, but it's really just 1 residue that is essential for stabilizing the structure of the finger. And so this is all nice, but the main question is, does it really matter to the activity of the protein, or is it just a nice exercise to understand the biophysics of these proteins? And so what Laura did actually was she measured the activity of the proteins, and she used a luciferase reporter assay to do that. And so in this particular case, she transfected a cell with a plasmid containing wild-type TTP, together with a plasmid containing the gene of Ranella luciferase that was fused to the 3' UTR of TNF-alpha, which is regulated actually by TTP. And so in the presence of TTP, you expect TTP to bind to the 3' UTR of TNF-alpha, and then TNF-alpha, that messenger mRNA gets degraded, and so less Ranella luciferase will be produced. And so by measuring the activity of the Ranella luciferase, we can actually have a sense of how much of the activity of TTPs. It's a bit of an indirect measurement for the activity, but it's, that's what has been used in the field. In addition, we also, so what Laura did was to measure the activity of wild-type TTP, and then of a version of TTP that had the tandem zinc finger domain of TC11D, where both zinc finger are folded. And what she observed is that actually the activity, so here you can see the first column is just the empty plasmid. So in that case, the activity of the Ranella luciferase is the highest, as expected. And then once you express TTP, the activity goes down, but you express TTP that has both zinc finger folded, the activity is actually quite a bit higher. It's about twice as high. And so what we learned from this study is that, TTP has somewhat evolved relatively recently to have the second zinc finger that folds upon binding, or is fully stabilized upon binding of RNA. And somehow that impacts the activity of the protein. That is, when it's like that, it's a little less active. So now I'm going to switch gear. I'm going to tell you a little more about other proteins. And we wanted to see if what we had learned on the human proteins was applicable to other proteins from this family. And so we focused on tandem zinc on proteins that contain this tandem zinc finger domain of the CCCH type, and quite a few actually are found in C. elegans, where they actually they're really important. Their function is really important during embryogenesis. And so at first we actually focused on 2 of these proteins, MEX5 and POS1. MEX5 and POS1, just to tell you a little bit about their function in the cell, is once the embryo is once the egg is fertilized to generate an embryo, one of the first things that happens is that an asymmetry is established in the cell, so that you can tell the head versus the tail. So an axis, an anterior posterior axis. And almost right after fertilization, one of the first events that happen is that MEX5 or MEX6, which is an homologous protein, they get concentrated on the anterior of the cell, and they get depleted in the posterior of the cell. And after this happens, other proteins, including POS1, get concentrated on the posterior of the cell. So this generation of a gradient of concentration in the cell is actually really important for the establishment and maintenance of this axis, anterior-posterior axis, because as you can see, if you have proteins that are segregated in different parts of the cell, upon cell division, this asymmetry is going to be maintained and the two daughter cells will be different. And eventually this will, generate different tissues. And in fact, all the tissues, in C. elegans are generated from, from these two cells. And so, so we were very interested in, in looking at the function of these proteins and how their RNA binding activity impacted, and their post-transcriptional regulation impacted their function in the cell. And so Asli Ertekin, she has been working in my lab, and she solved the structure of POS1 in the free state. And POS1, actually here you can see the structure is compared with that one of TC11D that is shown in gray. The one of TC11D is actually being solved when TC11D is bound to RNA. Asla solved the structure of POS1 in the free state. But the very first thing that you can see is that the structure are actually very similar to one another. Of course, the linker in POS1, it's very, it's very flexible, so she couldn't really get one unique structure. And so once she had the structure of the free POS1, she wanted to see if that could explain the different RNA binding specificity that was observed for POS1. In fact, Sean Ryder, who is also at UMass Medical School with his lab, they had determined the RNA binding specificity of POS1, and they had determined it to be different from that one of the human protein TTP and TC11D. And so here you can see at the bottom of this slide, the two different RNA binding specificity. And what you can observe is that actually the first half of the RNA sequence is the same, that is recognized by the second zinc finger in both TC11D and POS1. What is different is really the RNA binding specificity that is recognized by the first zinc finger, which is on the 3' end of this mRNA. But actually, just by looking at the structure, we couldn't really figure out what was so different that could explain the different RNA binding specificity. And so, but looking more in details, actually, Asla could see that the binding pocket, particularly for the first and the second, the binding pockets for the first and the second nucleotides in POS1 are a lot more shallow than in TC11D, and so this might explain why you can fit different nucleotides, although the interaction is strengthened through electrostatic interaction with arginine residue. So one thing that we still, though, when we were looking at the structure, we still couldn't figure out, because as you can see, there is more than 4 nucleotides, which is what the human proteins recognize, each zinc finger recognize 4 nucleotides. And so when Asla started to bind RNA to POS1, what she observed is that the spectrum effectively stayed the same, despite the fact that she was binding different RNA sequences. And so so from this, we gathered that when she was binding these RNA sequences, given the fact that the HSQC of POS1 is the same, she could infer that the POS1 was binding the same nucleotides. And so that indicated that some of the nucleotides from these sequences could flip out of the binding site, so that those that were actually exposed to the protein are the same despite this sequence. And so so these experiments actually were really useful for us to understand that, both zinc finger recognize still these 4 nucleotides, although in POS1 we can have more variability where we can actually see some nucleotide flipping out of the binding pockets. So then, given the fact that she had this kind of minimal sequence, she did a very thorough mutational studies, and she measured the difference in the free energy of binding. And what she observed is actually that the identity of a nucleotide that is bound in the first binding pocket impacts the nucleotide that can be bound in the second binding pocket, and vice versa. And so by, by putting all the data together, what she was able to determine is that there is some allosteric communication between the fourth binding pocket, particularly between the first and the second, and somewhat also the fourth binding pocket impacts what happens in the first two. And then also that you can get some of these nucleotide flipping out on the binding pocket. And so that might explain this, larger promiscuity, if you like, of the second zinc finger. So Asla also proceeded to characterize the dynamics of POS1. And what she observed is actually that in the fast timescale dynamics, so in the sub-nanosecond timescale, it's actually the two zinc fingers are very similar. You can see a similar profile. You can see this dip in the middle of the zinc finger, which is actually the second cysteines in both zinc finger. But one thing that was also, but generally the two zinc fingers are relatively stable. I mean, they're not two dynamics, except for the tail of the proteins, and of course the linker region. But one thing that was interesting that Asla observed is that every residue actually seemed to undergo fluctuations in the structural fluctuation in the microsecond to millisecond time scale. And so we actually use CPMG experiment to characterize these motions. And what Asla observed is that for zinc finger 1, so in both zinc finger, every residue in the zinc finger is undergoing a transition in a microsecond to millisecond time scale, and a two-state process was sufficient to identify the, characterize the exchange. But all the residues, but the residue in zinc finger 1 could be separated in two groups. One that is highlighted in the figure in gray that exchanges at a lower rate than the group in red. The probability of the two states are roughly the same. And then the histidine and the cysteine that coordinate the zinc, they seem to exchange at a faster rate that we couldn't quite characterize in the CPMG experiment. And so we were trying to, when we're trying to think what might be this second state that is at the equilibrium with the major state of the zinc finger, we thought that it could be unfolding. It could be that, given the fact that it's so global, they could be the unfolding of the protein. And so, if that is the case, if this hypothesis is correct, then we would expect the chemical shift that we measure in the CPMG relaxation dispersion experiment, they should correspond to the secondary shift. And, this is somewhat the case. We got a correlation coefficient of about I can't really read it now, but it's about 0.75. So it's not perfect, but it's not perfect, but it's, it could be that this is the process that we're observing. When we actually look at the second zinc finger, we actually observe that, again, the residues could be grouped. And we see in this particular case, 3 groups. They also seem to be, on the N terminus of the protein is in one group. The C terminus of the zinc finger is maybe in the other group. The zinc coordinating residue, they always seem to be on their own in this process. And here, the probability of the two states are always the same, that there are these 3 rates. But the correlation, once we looked at the correlation with the secondary shift, we see that the correlation is not as good as for zinc finger 1. But if we actually exclude the purple residues, the residues in the first group, those that exchange with the slowest rate, we actually get a much better correlation. So it is possible that these residues are, undergoing, they might go to an intermediate state that is not quite folded. So at this point, we really don't know what this, if the protein is fully folded, we need to follow up on these studies. But, if this is the case, if, the protein is always at the equilibrium with an unfolded state, even if it's a small percentage, that might impact the behavior of POS1 in the protein. In fact, one of the critical elements that makes POS1 segregate between the two sides of the protein is actually its diffusion coefficient that is different in the anterior and posterior of the cell. And so transition to an unfolded state might impact that behavior. So so Davide Tavella worked on the other proteins, the other protein of C. elegans we study, and that is MEX5. And MEX5 is like TTP. It's has only one finger that is stably bound in this, that stably binds zinc. In this case, is the is the second zinc finger, and the first zinc finger is again sampling. Is in a molten globule and doesn't tightly bind zinc. So what Davide solved the structure for the second zinc finger, and what he observed is that actually it's even less structured than both POS1 and TC11D. In fact, a very short helix that in the other two protein is 6 residues. In this case, it's just a 310 turn of, with just 3 residues. And so what he observed is that, when he looked at the dynamics by NMR, he actually sees that this protein, this zinc finger, is actually more flexible, not surprisingly, than POS1, and particularly in a region that is rich of glycines. He also ran molecular dynamic simulation that are represented here. The root mean square fluctuation that he observes is shown in black in this plot. And again, he recapitulated the same result, that the zinc finger is generally flexible, and particularly in this glycine-rich region. But once he ran the molecular dynamics simulation, what he observed is that, like for MEX5, like he did for TTP, he also observed the unfolding of MEX5 once he ran the simulation, and the unfolding was always due to some structural change that happens for the residues that were between the first and the second cysteine. So this seems to be a region that is critical for this protein to stabilize the structure of the zinc finger. And so he made actually a series of mutations trying to identify the residues that were essential for binding. And what he observed actually is, of all the mutations that he tried, none of them was able to either stabilize or destabilize unless he switched completely the two region. And so he was able, in this case, once he put all the residues that are found in zinc finger 2, which is stably structured, to in zinc finger 1, he observed the folding of that finger. So he also actually looked at the dynamics of the protein, and he sees also that MEX5 is sampling transition to a different conformational state in the microsecond to millisecond time scale. In this particular case, it seems like the finger globally moves as all the residues can be fitted globally, together at the same rate. But it doesn't seem like the unfolded state is the second state that is at the equilibrium with the major state, or at least the fully unfolded state. But we wanted to see, regardless, how stable MEX5 was. And so what we did was to look at the protein at different temperatures. And what Davide observed is that as he increased the temperature above 20 degrees Celsius, the probability for the second zinc finger to stably bind zinc decreased. In fact, the second zinc finger started to unfold. And when instead he went to temperatures that were lower than 10 degrees, he observed that actually the first zinc finger starts to fold. And so this is somewhat interesting because from his studies, what he gathered, in fact, at 7 degrees, he saw that the first zinc finger is approximately 30% folded. So what Davide observed is that the protein has one folded zinc finger and one not so structure, only in a very narrow range of temperature that is between 22 and 10 Celsius. And this is interesting because this is the same narrow range of temperature in which actually C. elegans is fertile. And I remind you that MEX5 is active when during embryogenesis. And so, the implication here is that maybe this protein has evolved to be in this state with one finger folded and one unfolded in this narrow range of temperature, which is when it needs to be active. Outside of this range, there hasn't been a lot of pressure to keep it in this state. And so as you go at lower temperature, when the animal is not fertile anymore, both zinc finger fold, and when you are higher temperature, or above 27 degrees, both zinc fingers unfold. And so with this, I end up, and I just want to thank everybody that was involved in this project. I think I mentioned them. I mentioned Laura Divo, Davide Tavella, and Asli Ertekin. And of course, Brittany Morgan, I didn't get the chance to discuss her work on, on this, on this project. And then of course, Sean Ryder, that has collaborated with us on the C. elegans proteins and NIH funding. And I'm happy to take any questions.
Thank you, Francesca. So again, if you want to ask a question, please type it into the Q&A. Don't type into the chat, or indicate by raising your hand that you want to be unmuted to ask a question. While people are typing, let me take the moderator's prerogative and ask you a couple questions. In the first part, does the threonine that you found is stabilizing, is it forming a classical end cap on that small helix?
It is. I guess it is. So it's from the side chain of the threonine to the C. So I guess it is an end cap. But actually, that motif is often found in membrane proteins. So once we realized that, I looked in the literature, and it's a very common motif that is found in membrane protein to introduce a kink in the helix. So often enough, that is what, the formation of this particular hydrogen bond introduces a kink in the helix. So I guess had I worked on membrane protein, I would have recognized it sooner.
So a question from Walter Chazen, does the second, back in about TTP, does the second finger bind zinc with high affinity in the presence of RNA?
Yes, yes. Although we haven't measured the RNA binding affinity, we do know that it's stable and it binds zinc stably, yes.
And then Rolf Bolins asks, what will a small pH change do to stability or unfolding of the fingers?
So that is a good question. I mean, one thing is that the histidine coordinates the zinc, so any change in pH is going to impact the stability of the finger as it changes the pH of the histidine. We haven't done, studies to see, how other, we haven't really done a study, a pH study, but I'm sure that it will impact.
And JD Paul asks, given this temperature range that you've noticed for formation of the fingers, does MEX5 have any role in a cold stress response?
So I am not sure in C. elegans in embryogenesis what happens in cold shock response. I do know that, as I said, the animal, the worm is infertile, and that is generally associated to changes in the metabolism that is really dramatically impacted by the temperature. But I don't know if MEX5 is involved in that.
And then a question from Fred Damberger, are there post-translational modifications that could be impacting function?
Post-translational, absolutely. So these proteins, they all get phosphorylated. They are phosphorylated outside the RNA binding domain, but they all get phosphorylated. So TTP, TC11D, and both POS1 and MEX5. So that certainly is the case, that they are, and their function is changes. So phosphorylation of TTP seems to inactivate TTP. In the case of POS1 and MEX5, phosphorylation impacts how they segregate in the cell. And in fact, that is a way, I mean, the current model is that phosphorylation impacts the ability, how this protein diffuse in the different part of the cells, and that is really what causes the gradient of concentration that is observed. So yes.
And a possible follow-up to that, There's a question about whether anyone has measured the translational diffusion rates of the folded or unfolded forms.
No, for the unfolded and folded form, no, they have not. They have, so the way they measure is, when they looked at the full length, so when they looked at the diffusion of the protein, they're looking at the full length protein, and they're generally, FRAP has been used also, fluorescence cross-correlation spectroscopy, and they generally are tagged with with a fluorophore. GFP in all cases. So that's how they have established this change in the diffusivity, if you like, of the protein.
Let's see, does the second unfolded finger have higher specificity for RNA?
No, in TTP, the zinc fingers seem to bind RNA. They bind RNA with the same sequence, although with the same specificity, although we have done a lot of studies, and I didn't present that data today, but what we see is that the protein bind and folds, although there is always a little bit of pre-folding, and then the protein binds. So because it binds and folds, if you have a sequence, so the zinc finger that is always folded always grab the RNA, the sequence that it, for which it has the higher affinity, and then TTP the second finger might fold on the sequence for which it has a lower affinity. I don't know if that made sense, but so the two zinc fingers, once they're folded, they have similar, RNA binding affinity, but because when it binds RNA, the second zinc finger is not fully folded, that might impact how the protein recognizes RNA.
Okay, I think we've gotten nearly all the questions, and in interest of time, I think we should move on to the second talk.
There was one question that was not answered because it was liked so much that it moved to the top.
Ah, okay.
I just read it. Very exciting talk. Did you try to denature ZF2 to see from the chemical shift difference if the relaxation excess state is unfolded state or not? Is the unfolded state or not?
So, we are in the process of doing that. So we can unfold it. We haven't assigned it yet. So that needs to be done. That's where
I had another question, maybe as a follow-up, which is you observe, your correlation between the relaxation dispersion derived chemical shifts and the denatured ones were dominated by 2 points or so, which were around 6 to...
Yeah, that's very high. Yeah.
Exactly. And I was just wondering, this is a huge effect. So do you know which ones these are? And can you explain why they have such a big shift?
So I have looked at that. I don't recall, to be honest, right now.
But there's an explanation.
I'm sorry, I can look. There is an explanation. I just don't remember it.
Okay. That happens to me all the time. Thank you.
Okay, so with that, thank you very much, Francesca. And then I'll turn things back over to Marcus.
Yeah, okay, so our...
or to May.
Our second speaker today is PK Madhu. It's a pleasure to have him with us. So Madhu obtained his PhD in physics at the India Institute of Science in Bangalore in 1997. And then he went to Europe to do postdoctoral research and also as a visiting scientist. So he was first at Weizmann Institute of Science with Shimon Vega for a couple of years, where he developed phase-modulated Lee Goldberg sequences for homonuclear decoupling and to get high-resolution proton spectra of solids. And then he went to Stockholm University and Southampton and worked with Malcolm Levitt and continued to develop a lot of NMR techniques with Malcolm, asymmetry-based pulse sequences for decoupling and maybe also recoupling. So he returned to India and started his own group at the Tata Institute of Fundamental Research in around 2003 and has continued to push the development of homonuclear and heteronuclear decoupling sequences in solids to higher and higher, fast and fast MAS. He has also, helped to refine redoor and dipshift type of techniques for fast MAS applications. So his group really has been at the forefront of NMR, solid state NMR method development, and his lab has also applied some of these methods to the study of A-beta oligomers. So today he's going to tell us about this heteronuclear decoupling techniques for all sorts of MAS frequencies, right? So welcome.
Thanks, Mike, for that fantastic introduction. Thanks also to Marcus, Christian, Lauren, and all the others for inviting me to be a part of this seminar series, which is indeed a great privilege to be over here. And I must say that I had a little bit of an apprehension after I said a yes to Marcus, because I realized that I don't have much of this biomolecular NMR at this point of time. So I told him that I would briefly talk a little bit about the heteronuclear spin decoupling. So he gave me a list of do's and don'ts. So I hope that I will sort of stick to his guidelines and make sure that the talk is satisfactory to everyone. So let me see how it goes. So what I'm going to do is to tell you a little bit about the heteronuclear spin decoupling, covering the mass frequencies which are commercially available. So that goes to till about 100 kilohertz to 120 kilohertz of that order. I mean, I may not have much of a spectrum in the very fast regime, but at least we will try to give you recipes for all possible regimes. So that's the whole idea behind this talk. Now, the reason for, of course, is one can have all these fantastic toys, as we also have, a 0.7 millimeter rotor, which can spin up to 111 kilohertz. Although I was told by one of my colleagues today that it probably doesn't spin more than 100 these days for various reasons. But nonetheless, we can do quite a lot of wonders with this. So if you look at the proton spectrum of adamantane, you can actually get the splitting of these two lines over here. But then Darren Bauer had demonstrated a couple of years ago that with homonuclear decoupling at 10 kilohertz, also you can get a similar spectrum. So one needs a little bit of the methodology knowledge to push the concept of solid state NMR, the magic angle spinning, the radio frequency pulses to get the right kind of information that the people have been presenting in this seminar series all along. So that will be sort of my background in presenting this spin decoupling. So as you know, the solid state NMR in the magic angle spinning regime has been touching all kind of fantastic inroads. I mean, there's a large range of systems which people have been working on, structural biology, the dynamics, and so on and so forth. And they have been made possible, as most of you know, and there have been talks on this in the last few weeks in this particular forum because of the higher magnetic fields, the higher magic angle spinning frequencies, better coils, for instance, the E-free probes, and so on and so forth, but also the techniques like the proton detection, for instance, which has been made possible with the higher MAS, and that boosts the sensitivity and also gives a leverage on the sample volume as well. But of course, one has to also look at the carbon-13 spectrum, the nitrogen-15, and that's where the heteronuclear spin decoupling comes in, despite the fact that you can still look at the protons at the higher MAS. But there are also the other regimes in which some of the proteins might behave better in terms of the sensitivity of the sample volume for that matter. So that will be roughly the focus of my talk. So I will talk about the heteronuclear spin decoupling, not so much on the homonuclear coupling, which Mike was alluding to. And we will also look at a little bit into the various aspects of the line width, the contributions to the line width which come from the homogeneous factors or the inhomogeneous factors. And there are several aspects to that which I will briefly allude to without going into much of the details. And people who are interested to get the details, there are these brilliant papers written by the group of Beat Meyer and Matthias Ernst. And there's a recent one in 2020, which also talks quite a lot about the very high magic angle spinning, about 150 kilohertz, and so on and so forth. So also for the benefit of many of the viewers, I was told that there's a large range of people from EPR to magnetic resonance solution NMR. There will be quite a few introductory slides, and then we will come to the crux of what I want to say. So that will be my major topic, the heteronuclear spin decoupling, the dipole-dipole coupling, in order to observe the carbon-13 and the nitrogen-15, and to improve the sensitivity and resolution of the various spectral techniques of involving the carbon-13 and the nitrogen-15s. And as you know, there are all kinds of these couplings present. So right from the hertz in the solution state to several tens or hundreds of or thousands of hertz in static solid. And then I know that there are the EPR people in the audience or the DNP people. I mean, they may be talking about the hyperfine splittings, which are even larger. So the question is, how do you manipulate these kind of interactions in a very clever way so that you get the spectrum that you want with a high resolution and also with the geometry parameters involved in it. And on top of the magnitude of these couplings, some of them may be anisotropic, they are orientation dependent, and they may be more tougher to handle with the standard Pulse sequences or with just the magic angle spinning. So that's something which we will try to see how to get rid of them if you want to get higher resolution and sensitivities. So the idea is pretty simple and is well known in the field of solution NMR. So if you have the two coupled lines, so all you have to do is to sort of have an RF irradiation on one of the spins. So basically we pulse on the I spin and look at the S spin, and that simple irradiation can be just a continuous wave pulse in the solution state and as well as in the solid state. And there are more sophisticated sequences for decoupling in the solution state, which are these Embler, Walls, Wurst, and so on and so forth. And that's the same sort of approach which the solid state people have also taken in the last 20 years or so. And there are excellent schemes in the solution state. So it's not just the sensitivity or the resolution which is important, also the bandwidth and the dependence to the offset and so on and so forth is important. So Wurst 40 over here, which is published by Eric Kupche and Ray Freeman, is one of the good sequences. I mean, it's got a fantastic bandwidth in this particular case. So that approach and that philosophy is quite the same in the solid state as well, where the idea is to go from a broad spectrum like this to a highly resolved spectrum like this, in the case of the carbon-13, which is coupled to proton. And essentially it has a large number of interactions which one has to worry about, the chemical shift and isotropy, the dipole coupling. It's not just the individual interactions, it's also the cross terms, it's the higher order terms, and so on and so forth. So what one has is the combination of the magic angle spinning and some suitably tailored pulse sequences which can give rise to such a high resolution spectrum. So that's the whole idea behind this talk. I mean, how does one design it, and what's the state of the art in this area? So again, those are the couplings, those are the magnitude of the couplings which one is dealing with. So typically you have something like 10 to 15 kilohertz in the case of the nitrogens coupled to the protons, and you have something like 25 kilohertz, the typical case, in the maximum case. And those are the couplings which we have to deal with or remove to get a high resolution spectra. So that's a schematic. I mean, you have some kind of a pulse sequence which is applied on the proton channel, and that removes these heteronuclear dipole couplings, these red arrows over here, and enables you to observe the carbon-13 as if it is not coupled to the neighboring protons. So that's what which we are trying to look at. So the obvious question which one can ask with all these developments is, why do you need such kind of a spin decoupling? If you have higher and higher magic angle spinning, can't that just do the job? So this is a slide again which I borrowed from Matthias Ernst. So this figure over here is a plot of the CH2 and the CH line widths in the samples of the glycine and the alanine, and that go as a function of the 1 over nu r, 1 over the magic angle spinning frequency as the theory predicts. And this is a spectra, this is a series of spectra of valine phenylalanine for a range of magic angle spinning frequencies without decoupling over here, and that's with decoupling at 48 kilohertz of magic angle spinning. That's pretty clear that even at these reasonably high magic angle spinning frequency of about 50 kilohertz, you still have to have some kind of a decoupling to get very highly resolved lines. And I will later, towards the end of the talk, show you that even at something like 80 kilohertz, 90 kilohertz, you still might have to apply some sensible decoupling to get a decent spectrum. And again, what people worried about in the past in developing these kind of sequences was the interference between the magic angle spinning and the radio frequency radiation. So both are time-dependent phenomena. So you can have the interference effects between both of these phenomena. There are various conditions which are good, and there are various conditions which are bad as well. And in special regimes, I mean, it's very difficult to find out which conditions are good or bad unless you know a little bit of the theory to handle such kind of resonance conditions as well. There are some standard conditions which are known, which are called the rotary resonance conditions or the Horner conditions, but there are many other conditions which also may have to be avoided, but there may be many other conditions which may be also excellent for the coupling. So it's also imperative to sort of understand the spin dynamics as much as possible to come up with good sequences. So that again is a very simplistic description of the Hamiltonians and the irradiation procedure in the case of a CW decoupling in solution. And that's pretty much the same for a CW decoupling in the solids as well. So this is again a spectrum of the carbon-13 which is coupled to the proton, and you have an RF irradiation which is given by a term like this. And what you're trying to remove is the scalar coupling, which is IZ, SZ, and get an average Hamiltonian, which looks like the chemical shift of the carbon-13 spin and the spin interaction term of the carbon-13. And that is pretty much the same in the case of solids. The Hamiltonian is again IZ, SZ term, which is pretty much like the scalar coupling. And all you do is to go into the interaction frame of the IZ with the application of the pulses, and you can prove that if you do a CW pulse, that goes to 0 to the first order. So that's all fine, actually. Now, that is not enough in most of the cases because of the presence of the higher order terms and the cross terms. And then, of course, as a function of the magic angle spinning, you can have other kind of effects, which again, Matthias Ernst has elaborated in great detail. The spin diffusion, for instance, where the culprit is actually the homonuclear dipole-dipole couplings among the protons. So even if you spin faster in that particular case, as you sort of average out these kind of couplings, you actually remove one of the beneficial factors which can aid the decoupling for the standard CW scheme. So the quality of the spectrum actually goes bad as you spin higher and higher in this particular case, which is sort of counterintuitive, but that has been extensively studied and elaborated by Matthias Ernst and others. So one of the schemes which actually sort of became the gold mark and paved the way for the later developments after the CW pulse sequence was the scheme called the two-pulse phase modulation, or the TPPM, which came from the group of Bob Griffin way back in '95. So in short, the TPPM is just a bunch of two pulses, as the name suggests, and it is phase modulated. It has got a phase of phi and a minus phi. And the duration of each of the pulse is close to 180 degrees. And the 2-phi ranges from 10 to 70 degrees. It depends upon the magic angle spinning. It depends upon the RF. The point is, it gives a fantastic spectrum of this particular example is the calcium formate carbon-13 spectrum, which is a broad line with the CW decoupling. It gives a sharp doublet, as you expect with the application of the TPPM. So that is fine. And later we will see that it does suffer from a few of the problems, but it is still one of the gold marks or the guideline of the development of many of the pulse sequences which later came around. So one thing which I want to notify is the number of parameters that one needs to optimize in many of these pulse sequences, and that is where we would like to give a recipe as to how to implement sequences without thinking too much about the optimal conditions. So here, as the name suggests, there are two parameters to optimize. So one is the pulse duration and the phase angle also, which could be dependent upon the RF and the magic angle spinning, as I said earlier on. So these are the issues which one needs to worry about in heteronuclear spin decoupling. So the goal is to get the better sensitivity and resolution with as simple an optimization as one would like to have, especially in the case of the signal restricted samples like membrane proteins or any other kind of samples for that matter. And the sequence should be less affected by the higher order terms and applicable over a very wide range of magic angle spinning frequencies. And, you know, pretty much the same kind of optimal conditions can we come up with some such scheme. So that's the whole question which people have been asking in the last few years. So there have been plenty of developments. I won't go into the details of that, but let me just sketch a few of those pulse sequences which came out after the TPPM was introduced, which obviously one thing which people did was to adapt the standard NMR techniques like supercycling the TPPM or phase modulated the TPPM or amplitude modulated, or kind of the standard things which people did in solution NMR to improve the robustness of a given pulse sequence. So you can sweep the frequency of the TPPM, and that's how it looks like, where the pulse duration is modulated. And you can supercycle that one with the conventional R bar, R kind of techniques. Spinal is again another technique which came which can also be related to the TPPM. And then there are other kind of sequences which came from the group of Tekili and Beatmeyer and Matthias Ernst, XIX, which is 2 pulses with phases of X and X bar. And the duration of the pulse is close to some multiples of the rotor period, which is 1 over the magic angle spinning frequency. You can supercycle that, and that becomes a Pissarro sequence, which came from the group of Tekili, Jeffrey Bodenhausen, and so on and so forth. And there have been many such variations. I'm not going into the full details of all these techniques. It suffices to say that with the TPPM, as I said earlier on, you get a good the resolution enhancement compared to the standard CW. And if you can sweep over this, the TPPM, like this, the frequency TPPM for that matter, you can also sweep the phase for that matter. The important thing which it gives is a robustness. The intensity of the resolution is perhaps the same as a very nicely optimized TPPM, but you have a much wider range of parameters to work with. You don't have to really worry about the most optimal conditions for the TPPM. You have a very wide range of it. So that's the robustness which all these modulations or the supercycling give you. So one thing which people looked at those times was whether these sequences can be extended to higher magic angle spinning. They can be, but there is no particular difference in the performance of any of these sequences. So there's a comparison of 6 of these decoupling sequences and the sample of histidine for that matter. And pretty much all of them behave the same fashion. So that probably says, or at least at that point of time said, that all the sequences beyond a certain frequency behaves pretty much in the same fashion, except for some robustness difference or some optimization ease, beyond which there is no big advantage. So I'll come back to this as we go on. And that's where another class of sequences came up from the group of Nielsen, which is called the RCW sequences. What he did was to put a 180-degree pulse in the middle of a CW scheme, and it is rotor synchronized. So the X pulse plus half of the pi pulse over here is rotor synchronized. And then there are different variations of these sequences. These are called the refocused continuous wave with different variations of A, B, C, D, E. So that's what is called the RCW sequence. And the rest of my talk, I will essentially concentrate on this particular class of sequence, but not in this particular way of implementation. So this way of implementation is rotor synchronized, and I will come back to it also. So that's where we were standing at some point of time, maybe 4 years ago, 5 years ago. So we had this continuous wave, which was improved considerably by the TPPM, and which gave rise to a large host of other sequences. And then we had the XIX and the Pissarro kind of sequences. And then we had this RCW kind of schemes where we had a refocusing 180-degree pulse. So that was fine. So before I go into the details of RCW and how one can improve that and how one can extend it to the higher MAs and the benefits, let me also point out that these sequences are important, not just in the context of the spectra which you get in the direct domain, which of course have an apparent line width, but there is also a refocusable line width, and that's what the various contributions to the line width sort of play a big role. So what is important is to sort of find out the effective transverse realization times. And it turns out that that's where there is a major difference between many of these pulse sequences, even at the higher spinning rate. As I said earlier, that at least in the direct dimension, all the pulse sequences at that point of time look pretty much the same. But now if you look at the T2 prime, they show a considerable difference. And another factor is the T2 prime also depends upon the RF power, actually. So if you have a high enough RF power, which of course you can put in all kinds of conditions, the value of the T2 prime you get is much longer than what you get in the low power decoupling at higher MAS. So conventionally what people believe is, or what people do is, and what is desirable is, if you spin fast enough, you do the second averaging with a low power decoupling. And if you spin low enough, you do the high power decoupling. But of course, there's a price you pay in terms of the transverse realization times and in terms of the spectral quality, which again will be highlighted in detail. So that's the fact of it for the sequences which I have listed over here. RCW is still not there. And what is the advantage of this long enough T2 prime comes in a series of experiments which people do pretty much for many applications in biomolecular samples. So one of them could be the inadequate. and most often could be the readout kind of sequences, the dip shift kind of sequences, where there's a whole bunch of echoes which is happening in the indirect domain. And the direct domain, you can actually look at the spectra which are decoupled. But what happens to the decoupling during this particular point of time is important. And it turns out that those T2 primes can go up to 100 milliseconds of that order, which is considerably long. And you get a very high sensitive spectrum in a standard frequency selective readout experiment, for example, in AB42. You can look at the selective coupling between certain residues. And because of this long transverse realization times, you can also measure relatively lower dipolar couplings as well. So that is the advantage which some of these sequences provide in echo-enabled experiments. So that is something which people might want to keep in mind. So let's just go back to the RCW decoupling scheme. So that's which Niels did a few years ago to improve the quality of decoupling by introducing a refocusing pulse, a pi pulse, in the middle of two CW blocks, and he just rotor-synchronized the X and half of Y to the rotor period. And that works fine. One of the caveat there was the pi pulse was supposed to have a very high RF power, which is not particularly desirable for many of these samples. And that's one caveat, but the major thing is that the principle is what I have sketched over here. So whatever is decaying in one half of the cycle gets refocused by the other half with the application of the pi pulse. So that's all fine. Now what is sort of subtle over here is this particular dip which happens in the rotor period. Now that is pretty important. So the dip actually became prominent when people started looking at these kind of sequences, not from the average Hamiltonian, but from a flood theory where one doesn't have to look at the performance of these sequences in a stroboscopic way. And then you can see that there are actually better conditions which are not in the rotor period, but which are slightly shifted away from the rotor periods. And there are plenty of other resonance conditions. So this is an intensity of one of the carbon resonances in a CH2 spin system, which is plotted as a function of the RF versus UR versus the tau A, which is the length of this particular CW block to the tau R. And there are resonance conditions by which we mean are the dip in the intensities. These are the bad conditions for decoupling. And they are similar to the XIX conditions, which have been extensively studied by the group in ETH, Vietmeyer, Matthias, and other people actually. So there are similarities between the RCW scheme and the XIAs, not just in the resonance conditions in terms of the limiting factors compared with the TPPM. So there are different cross terms which are responsible for the good performance or the bad performance of these schemes. So this is again what we realized, that it's better to implement these kind of schemes not in the rotor synchronized way, but in a little bit of a rotor asynchronized fashion. So it's a non-rotor synchronized of each CW block. So this is not an integer multiple of the rotor period, but slightly shifted. Now you can ask the question as to how much is that slight shift and whether it is a robust enough number so that people can just blindly put that number in for any arbitrary sample under any conditions of magic angle spinning and magnetic field. And turns out that you can do it, and that's what I will come to maybe in 2-3 slides. So from the performance point of view, although there were different versions of the RCW, it turns out that one of them is good enough, RCW-A is good enough, and it doesn't work very well at the synchronized conditions. It actually works much better either above the synchronized condition or below the synchronized condition. And the simple sequence A is already doing a much better job than all the other sophisticated schemes. So that's the good part of it. And this is another spectrum which actually shows the efficiency of these refocused CW schemes. So those are the responses, and they have been compared with a couple of schemes like the TPPM, for instance, and CW in a sample of history, and you can see that the RCW performs pretty well. The more than the performance factor in terms of intensity, what makes it robust is the forgiving nature of the pulse sequence with respect to the pulse flip angle. So it doesn't have to be necessarily 180 degrees the refocusing pulse. It can really cover a large range, and it's also fairly robust to the offset effects, much better than most of the other schemes in the literature. So those are the good part of it. So again, one can ask a question as to whether this is the end of it or can one improve it. Again, one can go back to the standard techniques in solutions to NMR or NMR in general. And you can think of it supercycling. The only thing is what kind of supercycling what one would love to have. So this is X, Y, X, and whether one should do an X bar, or Y bar, X bar. It turns out that it's a pretty bizarre sequence in that sense, which I don't quite understand actually. It turns out that it's not an X bar, Y bar, X bar, but it's actually an X bar, Y, X bar. So the Y is actually the same as this Y, and that seems to be doing an excellent job. And that is also clear from this particular plot, where it is the intensity is plotted as a function of the phase, and the phase, the theta and the phi. And it turns out that this particular phase of the refocusing pulse over here and here has to be the same, while the CW pulse has to be the R bar. So that's how this particular supercycling works. So it is not a real supercycling in the conventional sense of the RR bar, but this is what it does, and that does an excellent job actually. And that nature also we will see in terms of the robustness of different experimental parameters. So the sequence which comes about as a consequence of all this supercycling and this modification is something like this: X, Y, X, X bar, Y, X bar, where Y is a 180-degree pulse, and X is a CW pulse, which is almost equal to a rotor period. And what we found was that you just set it to something like 0.98. So 1 tau is bad. You can argue that you can go to 1.2 also, which is also fine. But we, as a habit, we have been doing this experiment by setting it to 0.98 of the tau, and it just works fantastic, irrespective of the sample, irrespective of the magnetic field, and the R forward till a particular regime of the magic angle spinning. And then, of course, you have to have another condition, which we will talk about in a couple of minutes. So that is one aspect of the RCW APA, or the supercycle RCWA, which we believe is one of the easiest schemes to optimize. So you really don't have to worry about anything. I mean, you just set the length of it to 0.98 of the rotor period, and that's pretty much you want to put it actually. And it works pretty well. So this is an example on one of the CH2 resonances, the glycine, at a magnetic field of 700 megahertz. The RF power is 90 kilohertz, which is the RF power of the CW block, as well as the RF power of the refocusing pulse. And the magic angle spinning is about 25 in this particular case. And you can see that the APA performs pretty well compared to most of the other sequences. So that's a good part of it. So there is an efficient decoupling. Well, I mean, we have also done it on certain protein samples. So this is an example from a GB1, which is compared with a well-optimized spinal, which is the red one. So you can see that the RCW APA, by just setting into that 0.98 of the tau, does a very good job. But spinal, of course, you have to optimize the pulse duration and the phase angles in a good way. The other aspect is, I mean, more than the spectral improvement, it is very tolerant towards the deviation from the optimal condition. So that's the tolerance to the deviation with respect to the RF, the rotor period deviations, and also from the flip angle of the refocusing pulse. There's fairly a robust sequence, which also makes it quite useful in unknown samples and unknown conditions. So now I come to the higher MAS side, which is, of course, the higher MAS, everything is a matter of definition. So what I mean is something which is beyond 40 kilohertz. So these are experiments which are done at 60 kilohertz, and later we will go into the 90 kilohertz as well. So what I have plotted over here is the response of the signal intensity is a numerical simulation of the CH2 resonance as a function of the flip angle of this particular pulse along the x-axis and the flip angle of the refocusing pulse along the y-axis for both the RCWA, the basic RCW sequence, and the supercycle version. And you can see that the APA is much better because there are very few bad conditions, which are these prominent ones, while here there are many such ones. But you can also see that they come at very systematic intervals or durations of the flip angles, something like odd multiples of 90 degrees in this particular case, and odd multiples of the 180 degrees in this particular case as well. That's important to note, and I will briefly highlight that. Well, over here is fairly robust, except for this particular condition, which should be avoided. And there are plenty of the slanting resonances. That I think that was a word which was coined by Matthias. Slanting resonance, which all have to be avoided. So it looks like a hopeless case if you look at here. There are plenty of such resonance conditions, but I will tell you the condition which one needs to adapt here for the best performance. Now, it's a good idea to understand as to what actually causes these bad conditions. So these are simulations done by switching off the homonuclear and heteronuclear dipole-dipole terms. And it turns out that it's the homonuclear and the heteronuclear cross terms which are causing these kind of resonance conditions. And this is a sequence, this is a figure in which the CSA is switched off. So this particular sequence is much prone to the homonuclear, heteronuclear dipole-dipole cross terms, where TPPM is of course much more prone to CSA dipolar cross terms. So those are the bare facts as far as these sequences are concerned. And the same kind of response can be mimicked in the experiment as well. And to cut the long story short, without going into the theoretical details, which is slightly important to understand the conditions, it suffices again to say that the good decoupling is always obtained when the cycle duration. So in this particular case, it is a cycle duration which is important, which is the entire length of these 6 pulses. And they should be the multiples of the power. And one can do a simple arithmetic and come up with a number that the sum n should be equal to 4 times the nu r by nu1. And it turns out that for the conditions, and I will sort of highlight it towards the end again, for the conditions that we took, like 60 kilohertz of spinning, 10 kilohertz of MAS, out of power, this is which one should adopt. And the good decoupling conditions come at about 22, 24, 26, which is the value of n. And that completely fixes the pulse duration and the cycle time. So that's a good part of this kind of decoupling, but you have to remember that it's now more of a flip angle dependence compared to the toward dependence in the low magic angle spinning regime. And again, if you take the slices of the spectrum, they compare pretty well. So the RCWAP again works out very well. So this is again examples from glycine ester, ethyl ester at 10 kilohertz of RF and the magic angle spinning at 60 and 50. And just been compared with different pulse sequences and all the conditions over here, but I will give you a take-home message in another 2-3 slides. And again, one can calculate the dephasing times and the offset dependence. And it turns out that the RCWAP again is slightly better than the other schemes, even under these kind of conditions. So that's all fine. Now, again, the question which one asks is there a simpler way of looking at all these schemes? And that's where we sort of came up with this unification idea of various decoupling schemes. And it turns out that all these pulses are just 2 pulses with a phase modulation. So all of them can be represented in a common language of a theta and a phi, or a relative phase difference between these two with a pulse duration of tau1 and a tau2. And depending upon the values of these thetas and the phis, you can have a CW, PPPM, XIX, RCW. So that's fairly simple enough to sort of visualize the evolution of all these decoupling schemes, and then you can actually find out the intensity of, again, a numerical simulation of some CH2 line width as a function of this phi and the timing. And the good part of it, you can recreate all the standard conditions like TPPM, which is over here, the XIX, which is over here, but you can also figure out newer and newer conditions, which could be important in some cases. And that is which we have done here. So there's a standard. These are responses to the standard pulse sequence, and that's a new kind of a sequence which came from this kind of an analysis, which we call it as a unified time and phase decoupling. You can do it at a high spin, you can do it at low spinning, and you can probably come up with better schemes, which we are still in the process of analyzing it. So this is a schematic of such a UTPD plot, an experimental, it's a simulated one. At the high MAS conditions, relatively higher MAS conditions and lower RF conditions. And one thing the take-home message of this particular slide is that the low phase of these decoupling sequences are tolerant to higher order effects of the dipole coupling. While if you go to the high phase, you get tolerant to the CSAs. So you can tune which decoupling sequences you might want to use depending upon the magnetic field that you're using or the spin system that you might be targeting. So that's again one aspect which comes out very well by the analysis of such a unified plot. So the previous one was the numerical simulation. The same kind of response comes about in the experimental factor as well. So this is again an example from a glycine ethyl ester at low RF and a relatively higher 50 magic angle spinning. And the relative phase difference is 30 degrees, which corresponds to sort of the TPPM regime. And this particular one corresponds to sort of the XIX regime, where the pulse durations become much longer and longer, but you can have a large number of good conditions to decouple very well. And they are also reasonably robust enough. So those are the stories, and this is a profile, a series of 1D spectra for some of the standard sequences. And this is, in particular, one of the unknown schemes on which we didn't know before, which we sort of extracted from the UTPD plot of a phase difference of 30 and the pulse durations corresponding to 60 and 40, which will add up to 360 degrees. In the TPPM, they actually add up to 180 degrees. So that's the only difference between these two spectra. And again, why the TPPM necessarily needs a 180-degree pulse is because it's again a question of averaging the scalar coupling. So it actually does a better averaging for the isotropic scalar couplings. And I won't again go into the details. The Foucault theory tells us that you can actually do these kind of plots of the coefficients of the spin interactions subjected to the RF. And it turns out that certain non-cyclic behavior is much more beneficial for a complete removal of many of the deleterious interactions, while cyclic ones may not be the best ones. But that's a matter of detail, but it's also important to keep that in mind. And the modulation, of course, helps on top of everything. And there has been a bunch of such sequences like the APA of RCW or XIX, which has been amplitude modulated, and supercycle as well. So all of them do a good job in terms of tolerance to the 1H offset and the J couplings to a large extent. Now, I just want to sort of sign off by comparing with the Waltz decoupling at the very high magic angle spinning. As Ishii sort of showed a while ago, the Waltz actually does a good job compared to some of the standard sequences. But what I'm going to show here is a spectrum which we collected, and just to adhere to the guidelines which Marcus said, these spectra are really from today, in fact, actually. Kaustup collected it right in the afternoon of today. And what he shows here is the comparison of the RCW on MLF compared with the Waltz 16, a 2 decoupling. There's not a big difference here, but there's a slight advantage if you stretch your imagination in terms of the RCW. The point is it's easier to optimize in that sense. You can look at the alpha resonance, the beta resonance, and the other resonances. They look nearly the same, but the optimization again is rather trivial for the RCW APA. And there are, of course, several other factors which I have not talked about, which of course give rise to the broadening, the technical issues, the sample preparation, and the higher order term, for instance. So that's the take-home message. If you haven't appreciated the earlier part of the talk, this is all what you need to set up the decoupling in the biomolecular samples. If you're sort of in the regime of up till about 35, 40 kilohertz, you might want to do this regime in which put in the sequence and put the tau A by tau R to 0.98, and that works out very well. If you're in this other regime, all you have to do is to fix these conditions. And these you know because you know the magic angle spinning, you know the RF power that you're putting in, and that gives you automatically the cycle time. And once I know the cycle time, I can calculate the tau Y from this particular relation. So the recipe is rather clear. There is nothing to optimize. So this is what we called it as a parameter independent optimization at the high MAS and the low RF. So that's the story of the spin decoupling as it stands now. And I acknowledge a few people who have been associated with this work. I see all along this work, along with Niels Nielsen. Shema did a little bit of the UTPD work towards the end, and who has been associated with decoupling for a long time. Vipin and Matthias also for lots of contributions to the Foucault theory in the RCW case, and Kaustup for collecting the spectra of the comparison between the Waltz 16 and the RCWA. I also acknowledge the experimental setup in the National Facility for High-Field NMR, the funding from the Department of Atomic Energy, TIFR, and the technical assistance from Manoj and Krishna. I finish it by flashing a conference slide of what Konstantin Ivanov, Daniela Bargil, Gerd will be organizing in August 26 to 28. So that's the timings of this conference, and you may register in this particular site. Thanks very much.
Thank you, Madhu, for a very interesting talk. So there are already several questions, but let me start with my own question. So the RCW sequence under fast MAS and with low power, with your rule, the cycle time would turn out to be on the order of half a millisecond or so if you try, one of these integer ratios of 24 or so. So I'm just wondering if you're meanwhile measuring some other couplings, maybe you're doing reador and you're trying this decoupling at the background. Have you seen anything unusual, or would this long cycle time have an effect on what you're trying to measure otherwise?
Yeah, it's actually Kaustubh has been one of my colleagues has been looking into some of these things in the last few days. So he has been doing those experiments at the high MAS and the low RF power. I don't think there is any deleterious effect in comparison with the other decoupling sequences. So the overall quality of the signal seems to be pretty good. There are interference effects because of the decoupling in the proton channel and the pulses on the carbon channel, the nitrogen channel, but that is kind of the same harmful effect. There is nothing special over there. So, or to put the story in a different way, so if you put the CW decoupling and the signal quality actually sort of improves. So I don't think we see any deleterious quality to that effect. Now, what we are also trying is to sort of see whether we can just use plain XIX, which actually corresponds to the beta Y going to 0, to see whether we can also get rid of the interference effects because of the pulses on the other channel. And I don't think so yet, but might work.
Yeah. So here's a question from anonymous attendee. Excellent talk. Does the improved RCW decoupling work during specific CP steps? Where you have 20 to 40 kilohertz CW on carbon and nitrogen. So it's kind of a little bit similar to my question. Yeah.
Mike, which question are you? This is the first question from anonymous attendee. Does the improved RCW decoupling work during specific? I would say it should anyway. Yeah. I mean, the RCW by itself, it's a standard decoupling sequence, which is probably even more robust in my opinion and easy to optimize. So it really should not matter.
Okay. So Zhe Honggan has a question for REDOR or HMQC. Spin echo intensity matters. Comments on decoupling sequence optimized for spin echo instead of the peak itself.
Yeah, I know. I mean, we just talked about it with one of my colleagues in the afternoon in this particular context. Yeah, if I understand the question correctly, so there might be an advantage of optimizing the sequences on the spin echo itself rather than in the direct domain of looking at the decoupled spectra. I would think it might be of an advantage. I can't say it with a complete confirmation, but this is something which we thought we would look into in the next couple of days or something like that. But there could be an advantage. Although, having said that, Sehong, the point is, as far as the RCW is concerned, there is really nothing to optimize in that sense. You know one of these parameters and you just put that in, and however we vary it, we hardly see any difference over there. But for the sequences like TPPM or the other sequences, maybe it is better to optimize it on the echo. Yeah, that would be my observation.
So here's another anonymous attendee question. Aren't the new this is jumping around, a new phase PD scheme just amplitude modulated TPPM? Is that true?
No, they are not amplitude modulated. I mean, you mean the low phase UTPD scheme? Well, the low phase UTPD scheme have different combinations, but the ones which I showed over here, the amplitude it's the same. It's just two pulses of the same amplitude. The phase difference matters. The frequency is modulated. The tau is modulated. It could be the unequal taus for that matter. So some of them would correspond to amplitude modulated TPPM aspects. There's a range of such sequences. We haven't looked at all of them, but some of them would correspond to the standard sequence. Some of them would correspond to the amplitude.
Okay.
That you're right.
Yeah. Next question is, I wonder, if you have tried other supercycle schemes for this decoupling sequence. Yeah,
if you're asking for the RCW, you're pretty much reaching the limit over there. I mean, if you look at the T2 primes, etc., you're already like 130 milliseconds, 140 milliseconds. And the same like what my Hong asked earlier, I mean, you're just complicating the sequence much more. You might gain a little bit, maybe 5% or something, but that's not worth the effort in my opinion. So you get it from the non-supercycle to the basic supercycle, there was a good difference in terms of robustness and in terms of the intensity. But beyond that, I wouldn't hardly think. In fact, we tried the R bar, R, and all kinds of stuff. it doesn't harm, but it doesn't improve considerably.
Mm-hmm. Okay. Here's a question. Next question is about, if you're spinning only at a 20 kilohertz, not very fast, what will happen if low RF decoupling is used? What's the difference from the fast MAS with low RF?
Oh, you will get nothing anyway, I think.
Yeah.
Yeah, maybe somebody else can comment, but you know, this is really a bad condition. And also this will be one of the resonance conditions, one of the bad resonance conditions.
Yeah.
So, you know, don't do that.
Yeah, Fred Dumberger asks, how do the different decoupling sequences compare with regard to B1 field inhomogeneity?
Where is the question? Okay, so I didn't talk about the B1 field inhomogeneity at all, and at least from my experiences, I haven't gone through that systematic calculation like Matthias Ernst was showing for the frequency switch leak, all that kind of sequences. At least in my reckoning, for the sequences that I tried with the same kind of setup, it sort of behaves in the same fashion, all of them being affected in the same way. I don't see a big difference, but that I have to sort of sound the caution, which we need to look at in more detail.
Okay.
Could I follow up quickly? I was just curious if you center pack your rotors in any way, or if they're fully packed.
We did that, you know, in the past for some of these sequences. We did that in the past, actually. For sub-frequency kind of a sequences, we did that. For the RCW, I'm not so sure whether we did that or not, but the CTPPM, we did that actually.
Because I mean,
it did improve a little bit compared to the normal ones, but my recollection is that it's not a tremendous improvement, but I have to go back and look at it.
Okay, because the conditions are quite close. It's 0.98, and
I know.
inhomogeneity could be.
I know that, yeah, I agree. I have to really get back to that. I can't tell you offhandedly.
Thanks a lot.
All right, so Kurt Zilm has the question, did you explore optimizing T2 for your A beta sample? You haven't talked about A beta, but you can tell us.
Yes, I mean, all we did, I mean, I can just tell you what we did. I mean, we didn't actually measure the T2 primes or anything in the A beta. That was all measured in some other samples. So we just took that particular decoupling sequence and put it onto the A beta. So we didn't try anything further to optimize it, because the signal intensity, although it was good enough, it is not really that fantastic to play around to optimize anyway. But whatever we could get it from glycine or some such sample worked very well for this. While with some of the standard decoupling sequences, we had to signal average for much longer time. So that was the advantage with RCW or the swept kind of sequences.
Okay. Last question from anonymous attendee is, do you see similar differences in T2 prime and line width for partially or fully deuterated samples under moderate to fast MAS? Also, have you tested using the optimized RCWA sequence to decouple C13 and 15 during proton detection?
Well, I myself haven't done the decoupling, the carbon 13 or the nitrogen 15. I have to ask my colleagues whether they've looked into that, but we haven't done it, and I don't see why it should not work. And the similar differences in the T2s and the line width, I would think you should get. I mean, yeah, that's what my feeling would be.
So you think it would be interesting to try. So Amir Goldberg asked another question. Do you expect any changes if you... I
would expect, Amir, I mean, yeah.
Nuclei.
I mean, we know that, you know, even for the RCW, I honestly haven't tried, but for the sweep frequency, we did get a big difference in the decoupling performance compared to TPPM or spinal. So that's something which I should have tried, but I haven't looked into that in detail, but we will do that.
So you have tried decoupling the quadruple nuclei from protons?
We have done it with the sweep frequency TPPM over spinal XIX and TPPM in the past, and there the sweep frequency was quite good actually.
Mm-hmm.
So we did that for the MQMAS kind of experiments and the STMAS kind of experiments, coupling it all the entire T1, and the response was quite excellent.
Okay, all right, that's good. Well, thank you. Thanks
very much.
Yeah. So I think our official session is over.